In this video, we will describe how to create an analysis template that can be saved, reused, modified, and exported using Glomax analysis software. Analysis templates define the curve fit type, data types, well naming, concentrations and dilution factors, and units that will be applied to the analysis of raw data from a plate read operation. Ultimately, these templates guide analysis and dictate what calculations will be applied to the data. Analysis templates can be created before or after data are collected. To create a new analysis template, touch the Results button on the main screen. Then, touch the Templates button. This will show you the list of existing templates and allow you to create a new template. Promega provides several example templates within the software to use as a starting point and to demonstrate the attributes of the different data analysis types. Touch the New Template button at the bottom of the screen. A blank template screen appears. Here you can select the type of curve fit. A linear standard curve allows you to specify samples with known concentration that define a linear standard curve, as well as samples with unknown concentration and blank wells. The concentration of unknown samples will be interpolated from the standard curve, and dilution factors will be applied if indicated. For example, this curve fit type would be used for quantifluor dye analysis. A linear response curve fit allows you to specify samples with known concentration. Data will be plotted assuming a linear relationship between concentration and data value. For example, this curve fit type would be used when performing a cell titration. A dose response EC50-IC50 curve fit allows you to specify samples with known concentration and plot data using a 4-parameter curve fit relationship between concentration and data value. For example, this curve fit type would be used when performing compound titrations with cells. When running a Promega nanobret assay, you can select the nanobret checkbox, which multiplies nanobret ratios by 1,000 to create millibret ratio units. The nanobret checkbox is only available with the Glomax Discover software. The Units text box allows you to enter units for the x-axis label on the data graph. Unit examples might include mgs per mil, micrograms, or moles. There are many unit possibilities, and the open text box allows you to enter whatever unit text is appropriate. In the center left side of the screen are buttons that allow you to specify the type of sample present in a well. Sample types could include knowns, unknowns, or blanks. When a curve fit is selected, the available options for well labeling will adjust to the options available for that curve fit type. In this example, we will build a template for linear standard curve analysis. To begin, we will assign some known wells. These wells contain samples of known concentration, also known as standards. Select the appropriate wells and touch the Known button. The selected wells turn yellow when assigned Known. All standards will be named the same, but we will assign a different concentration to each standard accordingly. Then we will select wells where the sample concentration is unknown and touch the button named Unknown. Notice that the selected wells turn green. We will give these a name and dilution factor for this type of well. Finally, we will select blank wells, which will be blanks for the experimental data set. Touch the button named Blank. These wells turn blue accordingly. Blank wells will be averaged and then subtracted from all known and unknown wells prior to analysis. When using software on a tablet, samples arrayed in non-adjacent wells can be selected by holding your finger in place on a well until a box appears. This enables multi-select. Next, tap any other additional wells as needed and assign well types accordingly. On a PC, simply use your mouse to select your well and holding down the Control or down arrow key, select other non-adjacent wells. Clear is used to clear all labels entered in case an error was made when creating the template. In order to be considered a complete data set, all known wells within a given treatment set or standard curve set must have the same name. For unknown wells, All wells of the same sample should have the same name, regardless of the dilution factor. Select all the wells for your experiment that will be analyzed together, blanks, unknowns, and knowns in this case. Then touch the well grouping button. The exclude button is used to exclude any given well that should be excluded from the analysis. When finished, touch the Save Template button at the bottom of the screen. This template can be applied to future data analysis, or it can be modified to create a new version of the template. The template can also be exported from the software and imported into Glomax software on another PC or tablet.